Welcome to our third podcast on the nervous system. And within this podcast, we are going to look at the larger units, principally of the central nervous system. We are going to look at the histologically distinct zones between the cerebrum, the cerebral cortex, as well as the spinal cord. And then lastly in this unit, we're going to look at the regeneration of nerves in the peripheral nervous system, as this does not occur to any large degree in the central nervous system. So as we begin a little review of spinal cord from anatomy and physiology, remember that the white matter comprised of myelinated axon is going to be in the periphery, whereas the gray matter, where we have neuronal cell bodies and unmyelinated fibers, are going to occur in the central portion. This is going to be somewhat of a butterfly-shaped region, or H-shaped as some call it. We're going to have the dorsal areas of the gray matter, which we call the dorsal horns, and the lower portion are the ventral horns. The dorsal horns are going to contain the axons coming in from sensory neurons. We'll also have interneurons in this location, and the ventral horns are going to be the cell bodies of our large motor neurons, most often of which are going to be of the multipolar type. So here we see an illustration depicting our dorsal root coming in. This is the dorsal surface of the spinal cord. This is the ventral surface. This dorsal root coming in is going to be our sensory fibers. This is going to be our dorsal horn of our gray matter. Here we have our ventral horn, and leaving we have the ventral root. Now this particular one talks about posterior and anterior. This is dorsal and this is ventral. Notice that both our dorsal sensory fibers and our ventral motor fibers come together to form a spinal nerve. Here we have a histological image of the spinal cord. Once again, we can see our gray matter in this H or butterfly shape. We have the dorsal horn, we have the ventral horn. This is the commissure that goes across from right to left sides. Fibers are passing back and forth. And then we have the white matter on the periphery. Another look of a section through the spinal cord. Very easy to identify the gray versus the white matter. And in this particular image, we've got a dorsal root ganglion. See, the fibers aren't directly connected, but this would be our dorsal root and fibers entering into the dorsal horn of the spinal cord. Now in the brain, this orientation is somewhat flipped. The white matter now is going to be deep or internal in the brain, whereas the gray matter is going to represent the cerebral cortex, and the cerebellar cortex is where we're going to find this gray matter cell bodies and unmyelinated fibers. In this image, you can see the gray on the outside, and we can see how we have increased the surface area of the gray matter with these indentations, these sulci, and these raised areas that are going to be the gyri of the brain. Now, both the brain and the spinal cord are going to be covered by fairly tough protective tissues. And so as we start on the outside and work our way in, the first is going to be the dura mater. This is the outer layer of our meninges, and essentially it's dense collagenous connective tissue. Now, this dura mater is extremely tough, and what we're going to see, especially associated with the skull, is we are going to have that portion associated with the periosteum, and so we have the periosteal layer, and then we're going to have the meningeal layer. This is the one that's going to be internal and deep and facing the actual tissue itself, and this is where we're going to see a lot of fibroblasts, connective tissue, as well as blood vessels. So here we have a illustration showing this cross section through the skull, the top of the head actually, and we can actually see scalp. We have epidermis, we have our hair on top. Here we see the actual bones, and as we look underneath, this is going to be the dura mater, this white area that's represented here. It is directly in contact with this inner table of the bone, of the flat bone, and that periosteal layer is going to be our dura mater. Now, when we look at the dura mater, it's going to extend from the brain down the brain stem and onto the spinal cord. And so our spinal dura is going to be this continuous tube of really tough connective tissue that's going to completely encase the spinal cord. And as you can see in this image, it's going to encase these proximal portions of our spinal nerves as well as our dorsal root ganglia. Now, if you've done the dissection in comparative anatomy where you looked at sheep's brains, you know some of them come in a very tough white envelope looking structure. Well, that's the dura mater, and you know you just are not going to rip that. You actually have to get scissors or a scalpel and cut through that. It gives you an idea of how intensely tough this material is. Now we have the dura mater, and this dura mater, directly beneath it, as we're moving deeper toward the spinal cord, we are going to have a subdural space. Now, this is a potential space between the dura mater and the next layer inside, which is the arachnoid, which we'll get to next. Potential space is much like we talked about in the lungs. It means it can exist, but physiologically and normally, the dura mater and the arachnoid are opposed against each other such that the space doesn't actually exist. You can separate them, you can pull them apart, but that doesn't naturally occur. So in this illustration, I believe this is through possibly a lamprey eel, and so what we have is our dorsal hollow nerve cord, and what we're going to be showing right here, this line that's running through, I think it's represented by number 3, that is going to be the spinal dura that's covering over our nerve cord. Now, of the 3 layers, the arachnoid is the middle layer, and it sounds a whole lot like arachnid, which is spider, because this area is going to be very loose. It's almost going to be web-like, and so that's where we get this name, the arachnoid layer. Now, while blood vessels do traverse through the arachnoid layer, for all intents and purposes, it is said to be avascular. We're not going to give off a lot of branches or a lot of sprouting. Some of the tissue will be supplied through some small blood vessels, but by and large, the arachnoid is a avascular tissue. Now, as we look at the arachnoid, it's primarily going to be a membrane, which of course is in contact with the dura mater. But then we're going to have these web-like trabeculators that are going to create this space, this subarachnoid space. And that is really where we end up with this name. As you can see in this illustration, we have the dura mater on top. We have our actual brain tissue here, the cerebral cortex, and here we have large blood vessels that are coursing through this space. And this is the subarachnoid space. The arachnoid layer is directly opposed to the dura mater, and you can see these arachnoid trabeculae creating this very web-like appearance. So in this illustration, we have our central nervous system tissue. As we're going to see, the pia mater is closely adhered to the brain. It's going to dive down into these sulci, and this web-like appearance that we see here, this is going to be our subarachnoid space, and it's very difficult to distinguish the arachnoid layer from the dura mater. This potentially is going to be the arachnoid. You see the darker line, and then we have the dura mater here. Very, very thick. Now, the innermost layer of the meninges is the pia mater, and when we were in anatomy, we learned that you could remove the dura, you could remove the arachnoid, but you could not remove the pia mater unless you were pulling off central nervous system tissue itself. It is directly adhered to the brain. And so what we see is we've got neuroglial cells that are playing a role here, connecting somewhat of the pia mater to the brain, but we also have fibroblasts that are going to be helping to adhere the pia and form it in especially the creases that we have in the brain. So as we look, again, we can see our scalp, we can see the bone, the flat bone, then we have the dura, which is the brown, we have the arachnoid membrane and the arachnoid space, and then this translucent glistening layer that they're trying to peel back, Excuse me. That is going to be the pia mater. And you can see how these blood vessels are flowing down through, underneath, and around. And so this is directly adhered to the brain tissue itself. In this histological image, we are looking at sections through the cerebellar cortex. And we'll learn how to distinguish that later on in this podcast. But as we can see, one of our sulci diving down, we can see this membrane that is running down through this, adhered to at least this side of the brain. This is going to be part of our pia mater, and that's how we can identify that. It's the only one that dives down into these sulci. Now, we mentioned before when we were talking about the microglial cells and the astrocytes and the other neuroglial cells, we mentioned the blood-brain barrier. And so here, as we look at the circulation that's coming to the central nervous system, not only are the astrocytes and their pedestals going to play a role in this, but the capillaries themselves also help to create the blood-brain barrier. These are going to be of the continuous type, which means between endothelial cells that comprise the capillaries, we're going to have fascia occludens, just like we saw in our epithelial cells. And so by and large, that's going to shut down diffusion that's going to happen, except for carbon dioxide and oxygen and some lipid-soluble molecules. But most of the rest of these molecules are going to be receptor-mediated transported. It's going to be facilitated in some way, either active or facilitated diffusion. Now that's going to be through the endothelial cells, but then with the astrocytes also having to screen this material, move this material between neurons in the bloodstream, those cells, the continuous capillary endothelial cells and the astrocytes, that accounts for our blood-brain barrier. Now the choroid plexus, we saw in a previous illustration that the pia mater does dive down into those sulci, but we're also going to have folds of pia mater in the ventricles of the brains, these open areas that we'll have inside. And in these ventricles of the brain, we have simple cuboidal cells that are going to produce cerebrospinal fluid. Now, ependymal cells that line the ventricles do this as well, but these choroid plexus cells are going to produce cerebrospinal fluid at a very rapid rate, maybe 14 to 36 mils per hour. And they produce at that rate because it's replaced 4 to 5 times a day as it's produced and removed, creating a tremendous circulation, not just to cushion the brain, but also as almost like, think of it like a moat around a castle. You got to cross that moat before you get to the castle to attack the people inside. Well, in this case, pathogens have to swim through this rapidly flowing cerebrospinal fluid to access central nervous system tissue. And that largely is going to be another very potent protective way of keeping the central nervous system somewhat immune separated from the rest of the body. And that includes the brain, brainstem, and the spinal cord. As you can see, the composition is primarily water, very little protein, but we're going to have sodium, potassium, chloride ions, all those ions you would expect to see in and around our neuronal cells, and again, principally present to be isotonic, but to create a rapid flowing environment. So now we're going to transition into actually looking at our organ histology somewhat. We're moving away from our tissues now and really starting to look at organs. We saw our neurons. Now we're going to put them together and to be able to histologically recognize whole organs based on the histology of the tissues that are present. So as I mentioned previously, cerebral cortex, this is the gray matter that surrounds the white matter of the brain, folded into gyri, and the sulci dive down in between, down deep into that cortical tissue. And we know our cerebral cortex, it's where we have learning, memory, informational analysis, initiation of motor response. And that's critical because our cerebellum is going to coordinate that before it actually goes out to our target muscles. And then also we've got the integration of sensory input so that we can make sense of what we're actually seeing. Now this is a very low magnification look at our cerebral cortex. We can see this fold that we can recognize on the outside as a gyri. Here's another gyri. And this indentation, the sulci that we have in our central nervous system tissue. The white matter is clearly visible, our myelinated fibers present here, and then our nerve cell bodies and unmyelinated processes on the outside. This is our gray matter of the cerebral cortex. Now, this is probably one of the most difficult sections other than blood cell development, I think, that we're going to go through this semester. It's very challenging to recognize these 6 layers in the central nervous system because they look very similar. So we're going to walk through this very closely and recognize zones as opposed to individual cells. Starting on the outer part, we've got the pia mater, and then the very first layer we're going to see is the molecular layer of the cortex. Then we're going to recognize, based on the size of cells and the number of cells that are present, granular and pyramidal layer. The molecular layer, the cells are very, very small. Few cells you're going to recognize. Mostly we're going to see processes, so you're not going to see a lot in the way of nuclei. The granular layer, the cell bodies are going to be fairly small and numerous, so it's going to look very granular. The pyramidal layer, this is where you're going to see very large cell bodies that are almost triangular or pyramid shaped. That's why we have that name pyramidal. Now notice we have external and internal granular and pyramidal layers. That is, as we're going deep, we're going to distinguish one from the other. And then lastly, just before we get to the white matter, we have a multiform layer which has cells of many different sizes and shapes, and so it's almost like the miscellaneous category in labeling our cells. This is an illustration trying to give us an appreciation of the different shapes and sizes of the cells that we have present in our 6 layers of the cerebral cortex. Out in the molecular layer, you can see principally fibers, processes, neurites, very few in the way of cell bodies. Then as we get into our granular layer, we're going to see small granule cells. Some of these are stellate cells, some are basket cells, but here you see these triangularly shaped pyramidal cells. That's where we have the P. So this is going to be our external granular, 3, external pyramidal. Then we have another granular layer, and you can see we may have some pyramidal cells in there as well, but the overall look is going to be more granular. So this is internal granular. Then we have these very large pyramidal cells in our internal pyramidal layer before we get to the last multiform layer, and then we have white matter beneath. Again, looking at illustrations, and you can see that in some they actually subdivide these. We're just going to stick with our 6, but you can see we've got very few, if any, in the way of cell bodies, so that's our molecular layer. You can see how these small cells are going to look very granular, tightly packed together. That's our external granular. Fewer cells, and you can see more of these pyramidal cells. That's our external pyramidal. Once again, we have the small, very numerous internal granular layer. Then we have our very large pyramidal cells, our internal pyramidal, multiform layer, and then we get into our white matter, which is beneath. Now, if it was that clear in actual histological preparations, it wouldn't be that big a deal. However, when you look at an actual section through cerebral cortex, it's somewhat challenging at best. I think that the molecular layer on the surface, because it's the first one, that's pretty easy. As we move our way down, finding the fibers and understanding where the white matter is, that's not bad, because then we'll get into the multiform layer, which is just above the white matter. You can see some of these large cell bodies in this region, so we know we're going to be in our internal pyramidal, which is adjacent to the multiform layer. Then it's these other 3 that we have to really begin to look at. Now, this is an extremely low magnification, and we're going to go in higher magnification and look at these in a little more detail as we move through this. All right, so here I've tried to put together sort of a lower mag view from the pia mater all the way down as far as we can see here. Here are our 6 layers. White matter is going to be off the bottom of the screen. And then this is the inset. We're going to blow the inset up over here. So We have our first layer, layer number 1, our molecular layer. And you see how small these cell bodies are and how few of them. We have a lot of fibers running through here as we compare this region to the underlying region. You can even see almost a demarcation line right there, distinguishing one from the other. So in the molecular layer, we have a lot of neuroglia, a lot of nerve terminal processes, and some of the cells that are called horizontal cells present in our molecular layer. Moving deep, we have the external granular layer. Moving deep, we have the external granular layer, which again are going to be comprised of these granule cells, stellate cells, and you can see they are much more tightly packed together. Many more of these, and they're going to appear granular in nature. And in some sections, it's really easy to see almost a dark haze here as opposed to a real light area in layer number 1. Now again, moving even deeper, we get into our external pyramidal layer, and here we can see these teardrop shaped, maybe think of them as spearhead shaped or triangular shaped. These are going to be our external pyramidal layer that we have. So that is 3 deep. Again, our small pyramidal cells, neuroglia. Neuroglia are going to be everywhere. They account for 80% of the mass that we have in our central nervous system. Then we get to our internal granular. So here we have layer 1. As we're going deep, you can see layer 2. Distinguish it from layer 1. Almost a lighter area in here. It seems like it might get a little darker, but again, we can somewhat see even here, those cells in that region are going to be those pyramidal cells. Then we get down here, the cells are going to be tightly packed together again, very small, numerous little tiny cell bodies. That's going to be our internal granular. And then even at the slow mag, you can see these large cells. Okay, these are going to be our, some call them bent cells, giant pyramidal neurons, but this is our internal pyramidal. This is where you're going to find the largest of those cells in this region. And then just below, we're going to have our multiform layer beneath. And then, again, white matter is even deep to that. So I think multiform, internal pyramidal, molecular, and the external granular, we can sort of associate those with each other, and then those middle two, we just have to look for the number of cells and then the shape of those small pyramidal cells to help us know where we actually are. Now, cerebellar cortex is much easier in the way of identification. A few layers, and they're going to really distinguish themselves from one another. But the functions of the cerebral cortex certainly maintain balance, equilibrium, muscle tone, but this is where we're going to have the coordination of our muscle movement, particularly for our skeletal muscles. Muscle memory is also going to be housed in the cerebellar cortex. You have athletes talk about being in the zone, not having to think, their body just reacts. That's because of the number of times they've done a particular movement and how that pattern of movements and that coordination of the movements has been stored in the cerebellar cortex. Now, in the cerebellum, we have 3 layers. We're going to have, as we saw before, we're going to have a molecular layer on the outside. We're going to have a granular layer, and this time it's going to be on the inside. And right along the boundary, we are going to have very large neuronal cells, and this is going to be the Purkinje cell layer. Now, Purkinje, not like we saw in the heart, but these are Purkinje cells in the central nervous system, and specifically in the cerebellar cortex. So our molecular layer comprised of all of these different cells, but predominantly we're going to be seeing a lot of processes of these cells, and that's why it looks molecular, especially compared to that bottom layer, the granular layer, where we have a ton of those small granular neuronal cell bodies. Now, I want to pay particular attention to layer 2, the Purkinje cell layer. These are going to be, they refer to large flask-like Purkinje cells, and they are going to be so prominent. And when you look at these cells, these are going to be the only cells that are going to transmit efferent information. Okay, these are the cells that are leaving the central nervous system, and these cells are always going to transmit inhibitory postsynaptic potentials. They're always going to be inhibitory in nature. And once you see these Purkinje cells, you will never forget them. You will never, this is one of the easiest ones that we have in our central nervous system. When you see this slide, you can see mostly processes, some small cell bodies in here. Then you see these large, humongous neuronal cells right along this layer lining up. That is our Purkinje layer. And then look at how granular this is on the inside. This is the granule layer. Here's a closer look at these Purkinje cells. Now this is upside down, but you would easily be able to recognize that. All the processes in the molecular layer, and then we have the granular layer. Actually, in this particular image, again, it's turned upside down. Now, the last thing that I wanted to mention in our nervous system unit, this is the last part of our third podcast and last podcast, but this is regeneration, and this is going to be in peripheral nerves, and this whole regeneration is called the axon reaction, but we can break it down into a couple different stages and different places. So at first, when you have an injury to a peripheral nerve, we're going to call this the local reaction. This is actually at the injury site. Let's say you have severed an axon. So what's going to happen is the damage ends, both the proximal and the distal portion of the axon, they're going to retract in both directions, and they're going to seal so that you don't lose any of that any more of the cytoplasmic material that is accumulated within. Now that material is going to continue to accumulate in the ends of these axons, and macrophages in the area, and to a lesser degree the Schwann cells, are going to remove any debris that might be in the immediate area. They're going to start to clean that up to get ready for the process of regeneration. Now the second stage, this is going to be the anterograde reaction, and this is going to be the port of the axon that is distal to the injury. And so remember, our anterograde movement is movement from the cell body to the axon terminus. So the first thing that's going to begin to happen is we're going to begin to degenerate that axon terminal. And by and large, we call this Wallerian degeneration or orthograde degeneration. We're going to be breaking down the axon and the myelin that cover that axon from the point of injury all the way toward the end, toward the target. But what's going to happen, remember, we have this basal lamina and the endoneurium. This has been surrounding the axon. And so the Schwann cells are going to form a Schwann cell tube within the space that was the place of our distal axon. So they're going to fill with that, and that basal lamina is going to maintain somewhat of a tunnel and a pathway with which we can attempt to regenerate this nerve process. Now, lastly, we have the retrograde reaction, and this is going to be toward the cell body end and that side. The cell body is going to hypertrophy. It's going to kick into high gear. And all those Nissl bodies and the nucleus, they're going to disperse. The nucleus is going to be displaced off to the side, and this is going to possibly take a couple of months. Because what's happening is the cell is gearing up for this repair process, and the axon is actually going to degenerate toward the cell body as far as that nearest collateral axon. And so we've severed that injury, and maybe we're going to retract a couple of millimeters, and when we get to that first collateral axon, that's where the degeneration is going to stop. And then at that point, we are going to start to sprout new axons, plural. They're going to begin to grow in every direction. However, only the axon process that finds the Schwann cell tube and reconnects with the target organ, only that is going to persist. The other sprouts will actually degenerate. And we can see that highlighted somewhat in this illustration. Here we have the injury to our axon. So there we have the injury. We are degenerating, so this is going to be our anterograde reaction. All right, so we got the Wallerian degeneration, orthograde degeneration. The cells are dying. The myelin sheaths are breaking down. Macrophages are coming and cleaning that up. And here we have our Schwann cell tube. Now, they're not showing such a great basal lamina here, but understand this is all somewhat within that basal lamina and surrounding that basal lamina tube. Now, our retrograde reaction, we're degenerating toward the first collateral. We'll say that's about in this location. We can see hypertrophy of our nerve cell bodies. The Nissl material has dispersed. The nucleus has been displaced off to the side. As again, we're shifting into synthesis gear. And now we're going to sprout several axon sprouts. However, you can see one of them is following this Schwann cell tube and is heading in the direction of our old axon terminal. Now, this is 3 months after the injury, and we have a successful regeneration. We formed a new synapsis, and now we have somewhat gotten back to our normal looking neuron. The Schwann cells have repopulated. They're forming the myelin sheaths. The accessory sprouts of the axons have been degenerated and are gone. And so we have made, in fact, that regeneration. But if we do not make contact, if we do not restore that axon terminal, then what we're going to have is we're basically going to have a disorganized axon growth, and an axon knot is going to form. We're going to have a cord of Schwann cells, and we will not be able to control that same target from that same nerve process.